Hello, and welcome to this presentation. This is the long version of my uh, talk for the Division F conference in Cambridge on um, the role of mass testing on a population level scale and its possible role in suppressing or even ending the current COVID-19 pandemic. This is joint work with UC Taipale, who's a biochemist at Cambridge, and Sten Linnerson, who's a molecular biologist at the Karolinska Institute. Right, so here's where we're going. The first third of the talk, I'll be talking about different types of testing for COVID-19 and the properties of those different tests. I'm certainly no expert in any of this, so this part will be like a summary or a news report from a mostly layperson's perspective, though I do hope that I'll be able to relay some of the more technical aspects of testing and the characteristics of different tests. The type of test we'll be talking about mostly is these rapid home tests that have been developed. They haven't been approved yet by the FDA in the US and other regulatory bodies, but they do exist. They are very cheap. They cost about $1 each, and they can be taken at home without any specialized equipment, much like pregnancy tests, and they just require a saliva sample. And the idea that I'll talk more about in a little bit is that if we all took these tests every day, we have a good chance at suppressing or even ending this pandemic altogether before a vaccine becomes available. Then a question that comes up is how much testing would be necessary for this to happen, and would this be possible? So in order to get a better understanding of this, we will consider two different and related epidemiological models that have been around for a while, and we will modify these models to include massive testing and see what the effect of that testing is. And that's the second part of the talk. And the third part of the talk will be actually our results. And I'll show some rigorous expressions that can quantify exactly the role of testing in large epidemics. Okay, so let's get started. First about testing. Why is testing so important? Well, one reason is that it allows us to perform the most basic task in infectious disease control. We can identify and separate infected and infectious individuals. It also helps calm fears in society that play a major role in disturbing and threatening the economy and public life at large. Testing also gives us, when done properly, the most accurate information that we can use to inform our models and make predictions about the evolution of an epidemic, decide public health policy, and perhaps most importantly from a scientific point of view, understanding the disease itself. Another specific issue to COVID-19, another issue which is specific to COVID-19 is that depending on who you ask, there are different studies that show that a very significant fraction of all infections are entirely asymptomatic, and those people can spread and do spread the disease, and the numbers that I've seen are anywhere between 20 and 70%. This whole spectrum, of course, is important wherever the actual number is, and it's only testing that can identify those asymptomatic spreaders and stop them. So clearly we need a very large number of tests if we can have any reasonable control of the pandemic, short of complete lockdowns that are apparently no longer realistic in most places. And the reason I want to describe tests carefully is that there are currently, in the US alone, more than 200 different FDA-approved tests, plus possibly another as many that have not yet been approved or are in development, and I'll try and describe some of those characteristics some of the characteristics of those tests. So the first type of test is an antibody test. This is the first type of test I'll be talking about. It's not relevant for us because its goal is to identify people that have been infected. This is a more difficult task than one would imagine, perhaps. because one reason is biochemistry itself, that antibodies from this novel coronavirus are very similar to antibodies caused by other viruses that have existed in the past, and it's not clear always exactly what antibodies are being detected. We also don't know the role of antibodies. Do they confer immunity? And how long is one immune after they've been infected and having recovered from the virus? The accuracy of these tests is not clear, like I mentioned, because of various reasons. And right now, there are more than 90 tests that have been allowed to circulate. Again, I'm mostly talking about the US because that's the largest market, but all these considerations apply broadly to most places in the world. And there are other social issues that may come up, that people might start using antibody test results as immunity passports, and they may start buying and selling them illegally and falsifying them and so on. But of course, they're important tests. They're likely to become much more common soon, but they won't enter our discussion from now on. The most common test that's being currently used to detect COVID-19 is the PCR test. These are molecular tests that are extremely accurate. They work extremely well. They're a small miracle of science. And they work like this schematic shows. First, you get a sample from either the pharynx or the nose of an individual. You extract the RNA of the virus that may be contained in that sample. You convert it to DNA that can be amplified, and then you use a PCR machine to amplify the genetic material in that sample, and then you look at the output. And if there is any virus, any significant amount of virus in the original sample, it would have been amplified enough to be detectable. And that's currently the gold standard, and it's a gold standard in a couple of different ways. One is that this is the only way, for example, in the US, for a COVID positive case to be counted as a COVID positive case. The only acceptable test is a PCR test. Another way that creates a bit of a problem to some of the considerations I'll be talking about in a minute is that every other candidate for a faster or cheaper test is compared against the PCR as a standard, and that causes problems. And I'll talk about that in a minute. But before that, let me just say that this in principle works only in a few hours. That means a few laboratory hours. And it is extremely sensitive. It almost never gives a false negative. In other words, it will almost always detect the virus if it's present in your system, even in very large quantities. It can detect 100 to 1,000 copies of viral RNA strands in just 1 milliliter of sample. Now, there are problems with these tests. One is that they're very slow. In most places, again, in the UK or in the US or other places in Europe, the turnaround times are slow, between 3 and 14 days in most places. And that has a very negative impact on policy, on public health policy, which is that by the time results come out, they're useless in terms of stopping the disease from spreading. Of course, they're very useful for treating the individual that has been tested, but if, say, someone gets infected, a few days go by, then they start getting mild symptoms, and they wait maybe a couple of days, then they take the test, comes back a few days later. By the time the test results come back, they are near the end of their infectious period of where they can spread the disease, or maybe even past that. So the speed is of the essence, and PCR doesn't do too well in that. There are also huge bottlenecks. There's a limit to how many PCR machines can be manufactured and bought by facilities. And even chemical reagents and lab supplies have been running low. There have been shortages observed, especially in areas where you get a surge in the epidemic. When you have big need for testing, that's when the testing program fails with PCR, because you get shortages. And also they're expensive, partly because they're over-regulated. Well, maybe if not over-regulated, they're extremely tightly regulated. The instruments, machines, and supplies that are used have to adhere to very specific rules. PCR testing requires specialized equipment, of course, and personnel. And there are also some social inequality issues that come up, again, not part of what we're interested in today, because a lot of people might take PCR tests privately, and if they cost, as they usually do, around 100 or 100 something dollars or pounds, then the poor can't take them and the rich can, and they get better care. Another minor issue that I will not discuss is that we can get more information from a PCR test result than just positive or negative. You can get an indication of how much viral load was initially present in the sample. And that's something that can be estimated based on the number of cycles that the amplification process took before the virus genetic material becomes visible. Those are not reported, but that's something that is beyond our considerations today. Now, there are a lot of possible ways one can try and fix those problems. One is group testing or pooling. That's when you pool a lot of samples together and test them together. And if they're negative, if the pooled sample is negative, then you know everybody was negative that has contributed to that sample. If it's positive, then you need to separate them and test them in smaller groups or individually and so on. That's a very useful and effective technique, especially for very rare diseases where you get very few real positives, or in the early stages of an epidemic. But at this point, it would save money if we did more group testing, more pooling, but it wouldn't really make a huge difference in terms of the other issues that are delays and shortages of tests. Then there are PCR-like tests. For example, instead of using a PCR machine, there's some somewhat cheaper machines that are also faster, including the Illumina sequencing machine, which is built for a completely different purpose for sequencing. It is faster, but it's still too slow, and it requires specialized equipment, and all samples need to be sent to a centralized location. Similarly, the University of Illinois at Urbana-Champaign has been in the news a lot recently. You may have heard. They introduced a saliva-based test. It was a modified PCR test, and they tested all incoming students and then all students every 2 weeks. That's a faster test. They used Yale's saliva direct test. That's a faster test. It's cheaper, but again, you may have seen in the news that there was a big surge of cases very quickly as soon as the campus opened, and they had to shut down again within 10 days or 2 weeks. And that clearly indicates that in places where there's a lot of activity and social contact, we need much, much more testing that happens faster. Now, there's a different type of test other than molecular PCR tests that's called an antigen test. An antigen test looks for something else, not for the SARS-CoV-2 RNA, not for the genetic material of the virus itself. It looks for an antigen. An antigen is just a fancy word that means anything that the test reacts to. So in this case, antigen tests detect not the virus RNA itself, but they detect certain proteins that are present in or on the virus. One example is a couple of tests that are very similar that have been built by 2 US companies. Here are their names. And what they detect is they detect a protein, the nucleocapsid protein, which is inside the virus in great quantities. And it can be detected using, again, a nasal or throat swab sample in much less time than a PCR test. It costs much less in terms of money, and it can be administered not in a big lab, but again, it requires specialized equipment. These 2 companies already have announced that they'll be making a lot of tests soon, but again, this doesn't give us enough testing capacity to be able to test at the scale of the entire population. One difficulty, as I mentioned, is that they need a proprietary reader. They need a specific machine that actually analyzes and detects the protein in the sample. And these companies can produce a lot of tests, but they can't produce a lot of machines to run these tests on at the same scale. And another problem is that, or not a problem, another characteristic of these tests is that because they look for this protein, which is actually inside the virus's membrane, they need to break it down and get into the virus. So that requires more chemicals, more reagents in order to be able to do that. Finally, the tests that are the main motivation for the work I'm talking about today are rapid tests that can be done at home. There are a lot of US-based companies. The one you are most likely to have heard about is E25 Bio, that make paper strip tests that are extremely cheap and detect COVID-19 based on a saliva sample. They cost as little as $1. You just spit on a paper strip that's about 1 by 5 inches long and wide, or even smaller than that. You put in a saline solution in a small cup and wait 15 minutes and get your results. The main difficulty that people have been having in getting these tests approved is that they're much less sensitive than PCR tests. This is an image that I showed before in connection with PCR, but didn't comment on it. Here you can see clearly the difference between the PCR test and a rapid test. So this axis is the days since someone gets exposed to the virus, and this is the viral load. It's in log scale because the viral load, that's why you see these straight lines. They look weird, but they're pretty close to being straight lines because both the multiplication of the virus in the body is exponential, and the way the quantity of the virus in your body decreases is also exponential after recovery. And this is the period when one is infectious and can infect others. And here we see that the PCR tests detect viral loads from this point higher. So it starts very early on, almost as soon as you get infected. Whereas the rapid test requires an order of magnitude more virus to be in your system before it can detect it. And because, as I said, the FDA compares everything to PCR tests, they look at this and they say, oh, that's not good enough. It's not nearly as sensitive, so we can't approve it. That is not necessarily a solid argument, and I'll say some more about it in a minute, but first let me mention a couple of things about this type of test. First of all, it doesn't detect the N protein, it detects the S protein, the spike protein that's on the surface of the virus, on the outside, and that's a very crucial protein in the virus's operation, in that that's the protein that binds with the ACE2 receptor on human epithelial cells, and that's how the virus gets into human cells and multiplies and causes COVID-19. And in that sense, well, I'm not going to go into any more of the biology into this, but I'll just say that although the sensitivity is only 60 to 80%, which means that only 60 to 80% of the time when you have someone who's really infected, you get a positive result, that's not so bad, because the only part you seem to miss is when they're not infectious. So you miss, compared to PCR, maybe a period of 1 day in the beginning, when someone has been infected, should be testing positive, but haven't started being infectious yet. And you miss the tail end where people have recovered, they're not infectious anymore, and the PCR, this tail can be much longer. It could be many more days or even weeks when someone has a lot of virus present in their system. It's mostly dead virus, and that's what the PCR detects for a long time after you're recovering. So these rapid tests are not as sensitive as PCR by a long shot, but that may not be so bad because they're not sensitive in a way that's not hurtful or harmful in how they spread the disease. You can think of this as an infectiousness test, not a disease test. So this basically says what I've just said in words. It's also put in writing here. PCR is much more sensitive, but that's okay in terms of trying to stop the virus, because if you want a serious clinical test for diagnostic purposes, you should get a PCR test. If you're testing every day just to stop infection from spreading, then you don't really lose anything or almost anything by doing these extremely simpler and cheaper rapid tests. Again, the FDA hasn't been approving them because it's comparing them to PCR, but there's discussion about possibly reframing these tests, not as diagnostic tests, but as transmission detecting tests or as simply surveillance tools. There has been some indication that the FDA might consider approving less sensitive tests if there's a broad high frequency testing plan for everybody. All right, so this suggestion has been gaining momentum, and there's a growing movement advocating that essentially everyone get tested every day at home. You know, you wake up in the morning and you brush your teeth, you take your antigen test, and then you go to work. If it's negative, everything's fine. If it's positive, then you isolate for 2 weeks. Or you take your test before every time you're about to have big social contact, before going to work or before going into a restaurant or the cinema or mass transit. You take your test and you only do that if it's negative. This website has a lot of information about this, rapidtest.org, and there's been a lot of activity especially in the last few weeks. One of the most vocal proponents of this idea is Michael Mina, a Harvard epidemiologist, but there's been also a lot of research been going on frantically since March. And in this context, the most relevant results are by people at Harvard and Yale, who claim that doing this not only can help significantly, but it can basically end the pandemic in a matter of weeks. It's not entirely obvious that this is a certainty, and others disagree, but there's very good indication that it's extremely helpful and a good idea to try and do it. And one important aspect of this plan is that if it's to work, only the government can do it. If you can't convince anybody to produce 5 billion tests unless they're certain that someone will actually buy them. So this needs centralized planning and control. And if we do this, it's not very hard to do the math. It turns out that even if, let's say, the US used 500 million tests per day, so everybody gets tested once and some people get tested twice, the cost of that, of the entire plan, would be less than 5% of the 3 trillion that have already been spent by Congress on supporting the economy because of COVID-related losses. So it's not so bad financially. There are concerns, like with every miracle plan. One is it possible to have such massive production capacity? It turns out that the materials are easy to obtain for these tests. It's actually the creating them that takes a long time or takes time. So that would have, for that to work, would have to enlist a lot of plants making them. Another question is, will people take the test? And if they do and they get a positive result, will they comply and isolate themselves? These are, of course, valid questions. Also, if we all do this at home and we don't go to a hospital or to a doctor, we lose a lot of important data because not everybody will be registered as a COVID case. So we lose a lot of public health surveillance data. And it may also, again, create a two-tier system where the rich take the super accurate PCR test and the poor take rapid tests. These are all concerns that are reasonable, but as I'll describe next, I don't think they're enough to stop someone from thinking that this is a plan with a good chance at working. So this is what motivated our work. We wanted to try and get precise about providing an answer to the following question: how much testing would be necessary for this plan to actually work? How big the frequency would need to be in order for the pandemic to be entirely suppressed just using testing? Right, so this is the second part of the talk. Now, what I'll describe is two simple epidemiological models. They're not entirely toy models, but they're not the most complex models either. Nevertheless, they're models that have been around for a while, and they're considered, or my understanding is that epidemiologists consider them useful. and relevant in some situations of diseases like COVID-19. So I'll first describe the models themselves, and then I will also explain how we add another layer of complexity to these models by including a mass testing protocol. Right. So here's a simple model. We have a population of n individuals. N is large. Initially, a proportion of them, a small proportion, mu n, are infected, and the rest are susceptible. And these individuals are connected in pairs in the following way. In the model, for every pair of individuals, while they connect them with probability alpha over n, or we don't connect them with probability 1 minus alpha over n. So everybody may possibly be connected with everybody else, and this creates a random network. And for large n, it's easy to see that this induces a very specific degree distribution. In other words, every person on the average has, not on the average, every person has a Poisson distribution of acquaintances or contacts on this graph with mean alpha. So this is our network that represents individuals' connections. And we assume that every individual who's infected, they stay infected for a random amount of time, which is exponentially distributed with a certain mean gamma. And we assume that the disease spreads by random contacts. How do random contacts work? So here we have, let's look at just this part of the graph where we have one infected individual who has just 3 contacts. That individual, during the time that they're infected, you can imagine that they run their own little Poisson process. It has rate beta times the number of their acquaintances, so 3 beta here in this example. And at the event times of this Poisson process, at each event time, they choose one of their acquaintances randomly, uniformly, and they infect them. And this goes on until the end of their infectious period, after which they become recovered, and they are immune or dead, and they do not play a role anymore in the spread of the epidemic. Hence the SIR name. I'm sure everybody's seen this a million times already, but SIR stands for susceptible, infected, recovered. And these are the 3 stages that individuals may be in during an epidemic. Right, so this is how the infection spreads. What we're also going to assume now is that in addition to this population moving around and having random contacts and getting infected and recovering and so on, there's an exogenous testing process which works as follows. It has a certain rate, theta times n, where n is the total size of the population. And during the event times of this Poisson process, at each event time, we randomly pick one of the people in the population and we test them. And if they're found negative, we don't do anything. If they're found positive, then we ask them to isolate for 2 weeks. Essentially, we just turn them immediately recovered. And there are some parameters associated with the testing. One is the sensitivity of the test. So there's a probability of false negative that hurts us, that slows down the control of the epidemic. We're also assuming that there are no false positives, that these tests have perfect specificity. This is clearly not a realistic assumption, but in a way it only makes our results, that we'll see in a minute, more conservative, because we assume that we don't quarantine anyone for no reason. And then we also allow for people not being compliant. So we allow for non-zero and significantly non-zero non-compliance probability when someone gets tested and gets a positive result. So if you think now about this whole process, you get your random graph, random connections, random infection times, and random contacts that infect others, and at the same time, random testing that stops these infections. This whole thing induces a high-dimensional Markov process where ST is the number of susceptible individuals times T, IT is the number of infected individuals, and RT is the number of recovered. And for large n, this model has this typical SIR behavior that you must have seen in the news at some point. This graph we've all seen many times. You see that initially almost everyone is susceptible, very few people are infected, and initially the epidemic rises exponentially. You get more and more infected people, fewer and fewer susceptible, but then the recovereds kick in, and eventually the epidemic dies out as there are no more infected people, no more susceptible people, and everybody's recovered. So this is a typical behavior you observe in these models. Now, there are, as I said, some are unrealistic assumptions. One is that the degree distribution is Poisson, that the distribution of our contacts is a random variable with Poisson distribution. This is not a realistic assumption, but that we'll fix in a minute with our next model. There are no false positives. An important issue is that we assume no geographical structure. This is a very hard issue to address, and very, very few models that are used in practice do incorporate geography and particular geometry in their modeling. We also don't include any characteristics specific to the disease. We just assume that someone gets infected and becomes immediately infectious. We don't account for incubation periods and so on. And we also assume something that's silly. We assume completely random testing. So we assume that you might wake up someone in the middle of the night when they're sleeping and just test them for no reason. Whereas targeted testing, which is what would be done in practice, would be intuitively much more effective. You wouldn't be testing someone, say, 3 times in an hour, which will likely happen with our model, but you'd be testing different people, and you'd be testing them right before they're about to possibly infect others. Right, so there are some unrealistic assumptions here. They're not incredibly simplistic, and most of them only make our results, the results that we'll see, more conservative. Before I show our results, I want to show one more model, which is a more general graph for the social connectivity of people. We may not want everyone's degree distribution to be Poisson. We may want to actually specify exactly how many acquaintances everyone will have in the population. So I may have my n individuals, I equals 1 through n, and for each one of them, I may specify exactly how many acquaintances they have, di. So di is the degree of node I, and it tells me that node I, or individual I, has di acquaintances. And I'm going to create a graph like that by first taking every individual and drawing half edges around them, as many as their degree. So if this individual has degree 2, then we draw 2 half edges. This individual has degree 1, we draw 1, and this individual has degree 5, so we draw 5 half edges. And then what we do is we just pick at random 2 half edges and connect them. So in this case, we pick these 2 perhaps, and we connect them, we join them, then we pick another 2, we join them, and we continue until we don't have any more left. And this is a very simple model. It's called the configuration model, or a random graph with fixed degree distribution. There's a possible problem that might occur with this construction, which is that I might connect myself to myself if I pick 2 edges that both emanate from me, or I may connect 2 individuals multiple times. So maybe I connect these 2 edges, and then I pick 2 edges again corresponding to these 2 individuals. So I might have multiple edges, but it's easy to fix this. You just repeat this random experiment until you get a simple graph with no self loops and no multiple edges. And if you don't like that, you can forget about this construction. I just described it because some people, including myself, find it easier to understand the model this way. You may just think of creating a random graph which is chosen uniformly among all graphs with a given degree sequence. So if you do this thing, then that's what you end up with. You end up with a graph which is randomly, uniformly chosen among all graphs with this degree sequence di. Now, in practice, we don't just specify an arbitrary degree sequence. Excuse me. What you really specify is a degree distribution. So you say, I want not a Poisson distribution, I want a heavy tail distribution. So I get, something like a self-similar graph or a scale-free graph or something else. So in general, you just specify a distribution pk, where pk is the proportion of individuals that have k edges. And then you select your di's so that the di's induce a distribution that's approximately close to pk. So you choose degrees so that the proportion of individuals with each fixed degree k is approximately pk. And you do this, you have to do this carefully. Now we're getting maybe a little bit too technical, but you have to do this both for the initially infected and the initially susceptible population. So once you fix the graph, then the epidemic and the testing happens exactly as in our previous Erdos-Renyi model. So you get infected individuals, they randomly connect with others and they randomly infect them. They stay infected for an exponential amount of time, and you have this outside process that randomly picks individuals in this population and tests them with these caveats that we saw before of perhaps false test results and non-compliance. Right, so again, our question is, how does the testing rate affect the evolution of the pandemic? Now I've described our motivation and I've described our models. Now I can tell you some of our results. So here's our first result. So let's get back to our simple model, the Erdos-Renyi model, or the Bernoulli random graph, as sometimes it's referred to in the epidemiology literature. So here, for a long time, it's been known how to compute the basic reproduction number for this epidemic. What are these constants here? Let me remind you, 1 over gamma is the average duration of the infectious period of an individual. Beta is the contact rate. So the higher the beta, the more often you make infectious contacts with others. And alpha is the degree of connectivity of the graph. It's the average number of individuals that each one is connected to. So without any testing for large graph, or as it gets large for a large population, the basic production number is this quantity. If you include testing like we described, this is how the basic reduction number changes. It changes by the addition of something in the denominator, which means, and this makes sense, of course, that it becomes smaller. So you affect the basic reproduction number by adding to the denominator this quantity, theta times 1 minus delta times q, where theta is the rate of the testing, q is the compliance probability, and 1 minus delta is the sensitivity of the test. It's the probability that you get a positive test result given that you're testing a positive individual. The proof of this is not very hard. The first part has been proved. I think these are certainly not the earliest references. These are just some very nice expositions that I found that I thought would be good to mention. At the end, I'll give full references to everything I've said, including the news articles about testing and so on. So the first part is known. The second part is based on a very simple observation. You may want to pause this video for a minute and think about this for yourself. The idea is that if you include testing, all you're doing is you're essentially shortening the effective infectious period of an individual. And you do this randomly and uniformly. In other words, by testing people at random times and entirely uniformly, all we're really doing is we're shortening the expected value, the mean duration of the infectiousness period. So gamma becomes gamma plus theta times 1 minus delta times q. Or in other words, the duration, the average duration of the infection, instead of 1 over gamma, is 1 over this, which is smaller. Right, so let's see what this looks like, because having such simple formulas, it's very tempting to plot them. So here we created a few plots with the following parameters. We assume that initially 1 in 10,000 individuals is infected, that the average number of acquaintances anyone has is 20. The average infectious period was taken to be 7 days, which is consistent with COVID-19. And we varied the contact rate so that we got different values of R0, of R0 for either different diseases, or you can think of this as the same disease with different strategies that are also being applied at the same time, like social distancing and so on. So if you have social distancing, or you close schools, or you force people to behave a certain way, you may decrease the average contact rate so that decreases R0. Now for the test, we assume that the test sensitivity is 70%. So there's a 30% chance, given that you're positive, that you'll get a negative result. We assume that the quarantine compliance is only 75%, and this seems to be consistent with empirical data on this. And theta is the average proportion of the population that you test per day. So this is what testing does. You start with an R0, let's say, which is 3, and the more you test, the faster it drops. In fact, the testing rate affects R0 in a convex way, which means that initially a little bit of testing makes a big difference, and later it makes a smaller difference, but it can always bring you below 1, which is where you want to be if the epidemic is to die out. Right, so this bottom curve corresponds to an epidemic with R0 being around 1.2, and testing, let's say, on the average, half the population every day, R0 immediately drops to half. And there's always a critical rate of testing that you want to be above. In other words, you want to test more than how much testing is needed in order for R0 to go below 1. And there's an explicit formula for that that's easy to compute. So as long as theta is bigger than theta star, where you have a very simple formula for that, then R0 of theta drops below 1. And here you see a plot. So notice here that theta star can be bigger than 1. So the proportion of the population that you're testing every day could be more than 1, meaning that, you know, this is random testing. We're not just selecting a group of people and testing them each day. We're just randomly testing people, and whenever this happens, there are going to be people that are not tested at all. There are people that are going to be tested multiple times each day. But on the average, this is just a rate. So if on the average you have 1.5 tests per day for every individual, then you can bring R0 of theta below 1, even for an extremely aggressive infectious disease with R0 equal to 5. Right. So this is encouraging. R0 is not the only measure of how an epidemic behaves. Another important measure is how many people eventually will have gotten sick because of the epidemic. So let's let capital T denote the total size of the epidemic. I'm not going to parse these formulas exactly. The important thing is that for a big population, as n goes to infinity, the proportion of the people that will have gotten infected is a certain number that we can compute that depends on R0. And the bigger the R0, the closer this number is to 1. And with random testing, we have, again, an explicit formula for this, that it's the same function, the same S, but now instead of R0, it depends on R0 of theta. The proof is along the same lines as for the previous theorem. And rather than going through the details, I think it's much easier to look at a diagram. Here we have the same parameters. We considered 10 different cases of 10 epidemics with exactly the same parameters as in the previous numerical example. And we look at the proportion of the population that will get infected by this epidemic as a function of the testing rate. So let's say that we look at this curve here. This corresponds to an epidemic with an R0 close to 1.2. And the more testing you include, the smaller the proportion of the people that will eventually get infected by the time the epidemic is done. And you see that all these lines at some point hit 0. So let's take the top line here, which would correspond to something like COVID-19 without any social distancing, without any measures, and without any testing. If we do nothing, then something like 96% of the population will get infected by the time the epidemic is over. But if we start and do testing, this comes down very quickly. So it comes down to about 50% by the time we do 55% of the people testing per day on the average, and it eventually comes down to 0. The point where these lines come down to 0 is exactly theta star, like the theta star we had before. This this is the critical value of the testing rate that brings R naught below 1. And this is the critical value of the testing rate that also makes the population, makes, I'm sorry, the epidemic disappear or vanish before it infects a non-negligible proportion of the population. The final thing that can happen is that if you start with very few infected individuals, remember in our assumptions for both our models, we assumed that there was a fraction of the population that was initially infected. If you start, instead of a fraction of the population, by a fixed number, small number of initially infected individuals, like 10 or 1,000 or even 10,000, then there's a small chance, but not a negligible chance, that you'll have a small epidemic. In other words, we won't have this behavior that we had before. We won't have that a significant proportion of the population will eventually get infected, but the epidemic will die out quickly. And again, I'm not going to read out the formula. The formula is the solution of this expression. If you set this equal to P and you solve for P, you get a probability, which is the probability that you'll get a small epidemic if you start with 1 infected individual. If you start with M infected individuals, the probability of having a small epidemic is P to the M. So we can compute this exactly, and this was computed by Martin Luff in '86. We can do the same computation in the case of testing, and we can get an explicit formula. Again, you can read this at your own time. I'll make all the slides available after the talk. And here's what these probabilities look like. So let's look at one of these lines. Again, it's the same range of parameters. So let's look at this bottom straight line. Actually, it's interesting that these are straight lines. It's not at all obvious if you look at these expressions that you get a linear solution, but it seems like you do. So what do you get? You get that if we start with just 1 infected individual and keep all other parameters the same in the epidemic as we had before, and the epidemic has an R naught of 3.5, the probability of having a small epidemic that will not affect the entire population, meaning that it will not infect a significant proportion of the population, is maybe 0.23. It's 23%. But if we test, the more we test, the more we increase the probability that we will contain this epidemic before it becomes a major epidemic. And at some point, if we do enough testing, we hit 1. So if you do enough testing, you can always, if you do enough testing early, you can always make sure that the epidemic is contained at an early stage and doesn't affect a significant proportion of the population at all. Again, the point where these probabilities hit 1 is theta star. So for each one of these, this point is where R naught of theta goes below 1. So if you do this much testing, if you do, say, 86% testing, meaning that every day on the average you test 86% of the population, or the number of tests you perform corresponds to approximately 86% of the population, then R naught drops below 1, and the probability of a small epidemic goes to 1. So the epidemic will be suppressed. Right, so these are our 3 results for the first model, for the random graph model with the Erdos-Renyi behavior. For the configuration model, there's a very similar result. I'll just talk about R naught. Again, the results are qualitatively identical and quantitatively very similar. There's a slightly more complicated formula for R naught without testing. Remember, before we had alpha, beta over betas and gamma. Now we have this quantity here that plays the role of alpha. And this is a function, this rather depends, it's a function null of the degree distribution, pk. This is for the configuration model where we fix the degree distribution of our random network for the epidemic. And the effect of testing is seen to be very much in the same form as before, in that a proportion of theta of the people being tested every day results in a smaller R naught, and the amount by which it is smaller is indicated by the fact that here you add something to the denominator. And if you just look at the graphs, they look very similar. I'm not going to go through all of these details. These are similar parameters like the ones we had before. The testing process has exactly the same parameters, and the epidemic evolution has very similar parameters, except for those describing the structure of the graph that are necessarily different since it's a different model. One cute thing to observe here is that this is, again, theta star. If you have a disease with a given R naught under these parameters, how much testing do you need to do for R naught to drop below 1? So if we have a disease, say, with R naught 2.5, we get here, so we need a bit less than 50% of the population being tested daily in order for the epidemic to be suppressed. The blue line is theta star for this model, and the dotted purple line, I hope you can see it, is the result we had before for the Edo-Shreni model. So although we have a very, very different type of graph here with a heavy tail degree distribution, very heavy tail for small k, like it behaves like 1 over k to the 1.75, and then it has an exponential cutoff around k equals 50. So this is a very, very different distribution than the Poisson. Nevertheless, the results we obtain are very similar. Right, so that's the end of our results. So what I wanted to say is that there is a proposal for suppressing this pandemic completely based on just testing. And this seems to be possible to do scientifically. It seems possible to do in terms of test production. And our epidemiological results suggest, with rigorous computations, that even if you do your testing completely at random, you will have very significant, very positive results. The basic reproduction number will decrease, the epidemic size will decrease, the benefits of testing are additive to those of other measures. In other words, you can do some social distancing and some testing. All of these things can work together. And testing every day may very well be the solution to containing or even ending the epidemic before a vaccine becomes available. You can look at the numbers again, but the bottom line is that here we see that in order for R naught to fall below 1, all we need to do is testing at the rate of about once per individual per day. So if we convince most people, most of the time before they enter some social situation, to take a test and not participate if they test positive, even with very, very imperfect tests, where we assume the sensitivity is only 70%, and with only 75% compliance, we can have very significant, a very significant impact. And this, of course, is now in the hands of politicians, but we thought it'd be nice to make some computations and offer some mathematical support for this argument. Finally, just a few words about what's been happening recently. If you've been following the news, rapid testing is all over the place. The US government announced a deal of $750 million buying rapid tests from Abbott Laboratories. Roche in Europe and the UK are making rapid tests available within the month. The Prime Minister in the UK announced an extremely ambitious program a couple of days ago with a cost of £100 billion. So there's a lot of traction that this thinking has been gaining, and it may well be that we stand a good chance to contain or suppress entirely this epidemic before a vaccine becomes widely available. That's all I had to say. Everything I talked about can be found in these links. References to all the mathy stuff and the news articles are given in that file. My slides are also publicly available, and we'll be posting our paper on my webpage over the next few days. Thank you very much.